Hi there, welcome to 1.15, which is all about cholesterol, HDLs, LDLs. The spec point is about you analysing and interpreting data on the possible significance for health of these, and the evidence for a causal relationship between cholesterol levels and cardiovascular disease. So it's kind of science linking them together, but most likely they're going to link it to you analysing some data, giving you tables and graphs, etc. So it's likely to be an analysis question, but obviously you need to know the science behind it before you can do that. So first of all, let's think about them giving you a table or a graph and asking you to describe the data, because it's often the first, part A of the question, describe the data. So we've come across this already in an earlier spec point. It does come up a lot. So if you're asked to describe a graph, you need to be, first of all, talking about the overall trend. So you need to say overall it increases then decreases, overall it increases then plateaus, overall it fluctuates and then decreases, whatever. Okay, so let's write down some of the words that you would be using. So you're going to talk about increase, decrease, plateau, that's when it goes flat, fluctuates when it goes up and down. Okay, so an overall trend, first of all right? Then you need to split it up into sections and you need to describe each section using numbers from the axes, okay? So you need to say that it increases from 10, blah, 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 up to 20, blah, blah, blah. Use the exact words that they use on the axes as well. Use the units. Make sure you use numbers in your quote. So it increases from this to this, then it fluctuates between here and here. Quote the numbers between what? Third thing is to do some kind of quantitative comparisons. So comparisons are words that usually end in So if you're comparing two lines, you could say one is higher than the other. You could say one is steeper than the other. You could say one is, this is where we get possibly another mark, using quantitative comparisons, twice as many, half as many, triple, a third, whatever. Okay, so you can see that some of them are just kind of descriptive, so things like higher, lower, faster, slower, but some of them are actually almost like calculations, twice as much, half as much, three times as much, a quarter of. They get you slightly more marks as well. And the final thing that we always do in a describe question is we need to do a calculation. And if I suggest that you always just do a percentage change, hopefully that will get you that mark. All right, so percentage change is the difference divided by the original times by 100. So let's have a look at an example of how we do one of these describe questions. So let's draw ourselves a little graph. Let's say you've got a graph that went up and plateaued like that. So the overall trend, apologies, mistake there. So your overall trend, I'm using my pen instead of the mouse. Overall trend, in this graph, you would say the graph shows that it increases and then plateaus. Next sentence. It increases from, and you'd quote the numbers from the axes. So you could say from 0 to 10, blah, 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 whatever it says in there. Remember to quote the units as well. Okay. From 0 up to 50, blah, 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 blah. Remember to quote the units as well. All right. Comparisons. So we could say time 10. So if we look here, we could do a comparison between time 10 and time 5, which would have been here. So if this was perhaps a rate, you could say that the rate at 10 seconds is twice that of at 5, because that would have been 25, wouldn't it? So at 10 seconds, it would have been twice however much it was at 5 seconds, because at 5 seconds it was 25 and at 10 seconds it was 50. So I've used a word to make a comparison here. The percentage change, the difference between 25 to 50 is 25. The original, the first one, is 25. Times it by 100. In that case, nice and easy, is 100%. So this shows a 100% increase from 5 seconds to 10 seconds. And that will be a perfect describe answer. Okay, so bear those points in mind when you're describing. The next kind of question that they might ask you is then to explain, which is a totally different question. So if we get a question with the command word explain, it means that you need to give possible reasons for something for why the correlation happens. So remember, graphs are correlation. They show that as this increases, that increases. As this increases, that decreases. As this increases, that plateaued. Explain means why does it do that? So why does it have that correlation? Use the science that you've learnt. It means that you need to use scientific step-by-step processes to explain that pattern. Why does that increase that? So that increases that because that activates that protein, and that protein binds to this, and that then binds to that, which causes this to happen. Yeah, it's a step-by-step process. Okay, so step-by-step scientific explanation. Okay, so use what you've learned in your course to explain how that links to that. How whatever it is on the x-axis links to the increase or decrease or whatever of that on the y-axis. Okay, so it's scientific explanation, scientific processes. All right, so explain questions are often reasons. Why does it have that correlation? You want a step-by-step scientific explanation, scientific process of how X causes Y. Okay, correlation is that as X increases, Y increases. Causation is that as X increases, it activates this protein which causes this enzyme to bind to this, blah, 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 which causes Y to increase. That's an explanation, okay? Very, very different. If the question asks you to conclude something, let's think about what they want you to talk about. So if they say conclude what the findings of this table or this graph show us, it really wants us to say the answer to the question we set out to investigate, okay, which in every experiment is about changing one variable, the independent, and keeping all the others the same, and seeing what happens to the dependent variable, okay? So answer the initial question, which always is going to be, let's move this over, what effect does changing the independent variable have on the dependent variable? And remember, your independent variable, if you want a graph, is always the x, and the dependent on a graph is always the y. If you're in a table, your independent is always the first column, and your dependent is always second column, or the last column if you've got lots of columns. Okay, so you can always get your dependent from your table or from your graph. They're always in the same places. Okay, and it's always when the controls are kept. And that's what gives us valid results, isn't it? Only changing the independent, keeping all of the other things, all the control variables, the same every time. Another thing they might ask you about is it something representative? Okay, now representative means that the characteristics of your sample are the same as the characteristics of the general population or of uh, the whole as a whole, whatever it is you're investigating. So the word for that is representative. Let's just make sure we understand all of these kind of working scientifically keywords. So representative means that sample shares the characteristics of the whole population. Okay, and the way you can make it more representative is by having a larger sample size. So in science, we always talk about using five uh, five of the independent variable, like five concentrations or five temperatures, etc. When we're talking about studies into people, which we are when we're talking about cardiovascular disease, we're talking about sample sizes, which are always better to be bigger. Okay, so just when you're thinking about numbers, when you're thinking about experiments in the lab, the number you're always thinking is five different independent variables. When we're talking about sample studies, we've got another number to think about, which is our sample size. And the larger your sample size is, the more representative it's going to be, and therefore the better the results are going to be. Right, now let's start to think about the science behind this spec point. So the spec point's about cholesterol. So it's about kind of analysing information about cholesterol, why we've talked about lots of those kind of keywords. But now let's talk about the real science of it. So let's start off with cholesterol. So I think when we say cholesterol, a lot of people just think, oh, fat. Without really knowing much more than that, which is totally understandable. But let's have a little look at some more of it. So you're right in that cholesterol is a type of fat, and we call the fats as a group lipids. So it's a type of lipid, right, which is why the spectrum comes after the triglyceride one. Why do we need cholesterol in our body? Now, the place that you've specifically come across it is in the phospholipid bilayers. Okay, so uses the phospholipid bilayer, which is in all the cell membranes, the plasma membranes around every cell. So you come across it there. It forms a large part of the sex hormones, which is like your estrogen and progesterone. We need it for vitamin D production. So we're getting a bit off spec now, but let's just make sure we're aware of what it does. And it's also used to repair cell walls. So there's a few jobs of cholesterol. Don't worry about learning them too much, but make sure you definitely know it's part of the phospholipid bilayer. Now we've got two other molecules that we need to know about, and these are called HDLs and LDLs. So HDL, and LDL. I'll come to LDL in a minute. So HDL stands for high density lipoprotein. HDL. And I'll just copy this box to do the equivalent, which is called LDL. Over here. Stands for low density lipoprotein. So we've got two molecules, HDL and LDL. Now, some textbooks and teachers, etc., simplify this and say that HDLs and LDLs are types of cholesterol. You actually need to know slightly more than that. They're not types of cholesterol. They're molecules that carry cholesterol. So when they're holding cholesterol, yes, they are kind of a type of cholesterol because they contain cholesterol, but really they're a molecule that's carrying cholesterol around. They're almost like a cholesterol taxi. So they carry the cholesterol, and one of them carries the cholesterol one way, and the other one carries the cholesterol the other way. So one of them will increase blood cholesterol, and one of them will decrease blood cholesterol. So the HDL, the high density lipoprotein, is the one that decreases blood cholesterol, and the LDL is the one that increases blood cholesterol. Now, when we're thinking about which one's good and which one's bad, it does, to a large extent, rely on where in the world we're talking about. So, in our society, a relatively rich society where we have access to lots of food and also to a lot of fatty food, generally we all have plenty of cholesterol. So our cell membranes have plenty of cholesterol. We have plenty of cholesterol to make our sex hormones, etc. So for us, we would say that the HDLs are the good ones. Because we want to decrease our blood cholesterol. We have plenty of cholesterol in our bodies, plenty of cholesterol in our diets, most of us. And so it's good that we have HDLs to decrease our blood cholesterol so it doesn't get too high, so it doesn't end up in our artery walls causing those plaques, those atheromas, leading to atherosclerosis. On the other hand, the LDLs increase your blood cholesterol, which for us in our society is a bad thing because we've already got so much cholesterol, we really don't need any more. It could start to be a risk factor. Obviously, if you were living somewhere where you didn't have access to lots of fatty food, for you, actually the LDLs are really essential to bring the cholesterol into your blood so it can get to your cells and build your phospholipid bilayers in your cells. Um, so that good and bad is a little bit subjective, if you like. Well, location and person specific. But for the majority of people, if we say HDLs, I say in my mind, not in an exam, H for happy, good, decreases blood cholesterol, and L, low, sad. Okay? They're the bad ones. They bring too much cholesterol into our blood. That's a bad thing. That's a danger. That's a risk factor for CVD. All right. That's the way I remember it, just as a little prompt of which one is which. So, in terms of someone that has plenty of cholesterol, the HDLs are decreasing our blood cholesterol and therefore decreasing the risk of cardiovascular disease. On the other hand, the LDLs are increasing the blood cholesterol, and any excess cholesterol may end up in the artery wall, may end up forming a plaque, an atheroma, atherosclerosis, etc. So, therefore, is increasing the risk of cardiovascular disease. If you've got high HDLs, therefore, that's going to be a good thing. If you've got high LDLs, that's a bad thing. Now, we all have both. We all have HDLs and LDLs, but it's the ratio between the two that's going to make a difference. And that ratio is partly genetic. Um, let's just make sure then that we've got a record of how this happens, because if you were given a graph in your exam that had on the x-axis LDL to HDL ratio, for example, and up the side incidence of cardiovascular disease, and it showed a correlation of increased LDLs and increased cardiovascular disease. That's a correlation. If the question then asks you to explain that, you need to give an explanation of how more LDLs increases your risk of cardiovascular disease. Now, it could be that they give you a second graph where it's got the amount of cholesterol and the incidence of cardiovascular disease, and it wants you to talk about both of them because both of them are going to play a part. More cholesterol, there's more to get in the walls. More more LDLs, there's more of these little taxis to carry that cholesterol to the artery walls. Okay, so both of them could play a part. All right. So if we've got more cholesterol, actually, I might just write this at the bottom because it's a little bit narrow there. If we've got more cholesterol, greater, If you've got more cholesterol, there is more to be accumulated in the artery wall. Remember how I always have to say in the artery wall, not just in the artery, with the really specific language, to form a plaque. There's a risk of atheroma, which is obviously called atherosclerosis. Okay, and if we have more LDL, they carry more cholesterol to the arteries. More cholesterol accumulates in the artery wall and then as above. Remember then, once you've got the atheroma in the artery wall, if the endothelium gets damaged, let's carry this on the next one. There. So if the endothelium gets damaged, it's going to trigger the blood clotting state. So remember that means it's going to release thromboplastin, and that's going to catalyze the breakdown of those molecules from the longer names to the shorter names, which ultimately results in that fibrin, which causes the mesh and the red blood cells and the platelets stick to it, and that can cause your blood clot. So trigger the blood clotting cascade, and then the blood clot and block artery further. It's already been narrowed from this one. That narrows the artery. Blocks the artery further, so less blood to get through, so less oxygen. So the cells respire less, and the cells may even die. And if that's your coronary artery, it's going to cause a heart attack. The fancy name for that is a myocardial infarction. If that happens in the brain, was the diagram to make sure we understand what's happening. So in our blood vessel, you've got cholesterol, and you've also got these little carriers called LDLs. So the LDLs, if you're visual, you'll find this a little bit easier. The LDLs carry the cholesterol to the blood vessel. Now, what may happen is we may get a plaque forming here. And one of the things that that plaque is made of is cholesterol. So this is called a plaque. One of the things that that's made of is cholesterol. So the more cholesterol, the more LDLs, the more risk we've got of getting this plaque. If then this surface here is damaged, then we release thromboplastin and we start the blood clotting cascade, and we might form a clot on here. Sometimes that clot could even dislodge like this, block the artery completely. Okay, what happens is that fibrin from the blood clotting cascade attracts the red blood cells and the platelets to get stuck on it, and it forms this big mass. It's blocking our arteries. We need the blood to get through, carrying the oxygen to the cells for respiration. But look, is getting blocked. And that's why it's all a risk factor. All right. So that's the end of that one.